I'm a doctoral candidate at Johns Hopkins University in the DocCo Chem Lab, and I'm excited to describe some of our recent work on instrumented microphysiological analytic platforms for precision measurement and manipulation of tissue functions. So here are DocCo's financial disclosures. Unfortunately, as a graduate student, I have none. So our laboratory research focuses on development of microphysiological models of human organs. Whoops, let me get the laser pointer working. Perfect. So we use multi-scale biofabrication techniques such as micro and nano biomaterials and fabrication, microfluidic organ-on-a-chip systems, and 3D bioprinting techniques. Using these bioengineered human tissue models, we aim to study human disease biology and perform predictive preclinical drug screening for cancer, cardiovascular disease, and neuromuscular diseases. So there are still several challenges that need to be fully overcome for the field to embrace microphysiological systems that we aim to kind of address. First, throughput needs to be increased. Second, longitudinal, non-invasive sample assessment needs to be realized, as sacrificing samples to assess endpoints is not sustainable. Finally, there should be a transition to automated fabrication, as manual processes for tissue chip fabrication lead to increased fabrication times and greater user-to-user variability. These are all problems we're aiming to address. So our group has been developing high-throughput engineered human heart tissue platforms for drug screening applications. Our EHT platform consists of human-induced pluripotent stem cell-derived cardiomyocytes and stromal cells seated within a hydrogel biomaterial scaffold and cast around 2 silicone posts, in which the deflection of the flexible posts, you can see here, allows us to measure contractile kinetics of our tissues. With our collaborator, Nathan Sniadecki at University of Washington, we modified the EHT platform by incorporating this magnet you can see here in the flexible post. These magnets, when coupled with an array of giant magnetoresistive or GMR sensors, allow for the measurement of post deflection in an automated and real-time manner. As posts are deflected, the resulting change in the magnetic field is detected by the sensors, converted into a voltage, and then can be used to calculate these twitch forces without expensive microscope-based methods, which are slow and low throughput. Our functional tissue chip assay technology enables a wide variety of tissue contractile kinetic measurements, as you can see down here, and it's been licensed to a biotech startup company, Carry Bio, for commercialization. Nick, I think you're giving a talk on this tomorrow, so if you want some selfless self-promotion here, what time's your talk?
10:15 a.m. It'll be the best talk of the day.
That's because mine's today. So previous work has identified mechanical actuation as a method for improving cardiac tissue maturation and enables research into a wide range of interesting mechanobiology questions. These studies have been largely limited by low throughput platforms. So we've also started to utilize our previous platform that we just mentioned, this high throughput magnet-based platform for measuring contractile function, to also enable research into magnetic actuation. Where we can use a magnetic coil to displace the magnet in our flexible post, pulling on the tissue. So here's a schematic illustration of our high-throughput magnetic actuation system. It consists of 3 parts. We have our array of posts. We have a magnet field forming unit for exercising the tissue, and underneath we have the monitoring unit, either magnet-based or imaging, depending on the experiments we're doing. Simulated and experimental results indicate that the system is able to apply uniform magnetic fields across the entire plate with a uniformity of about 95 to 96%. This allows us to apply nearly identical forces to all plate posts in this plate. So here we show the working principles for mechanical actuation in these engineered tissues. The magnetic posts deflection and periodic motion can be adjusted by controlling 3 parameters, namely the intensity of the field, the direction of the field, and the frequency of the field. On the right, you can see the deflection increases with intensity and direction of the magnetic fields. Next, we wanted to further increase throughput of our sensing and actuation platform. Our previous platform, commercialized with Carry Bio and developed in-house, as you can see over here, is only medium throughput and requires a larger number of cells and reagents. So working with Carry Bio, we aim to next adapt this into a 96-well plate platform and decrease the amount of reagents and cells required for each tissue. So here on the left, we show a schematic illustration of the 96-well plate platform. And one of the problems we identified very early on in the design phase was that skeletal muscle exerts much greater forces during contraction and have higher passive force than cardiac tissues, resulting in permanent deflection of the post. And this causes the post, when we pace these tissues and cause contraction, the post deflect beyond their linear elastic region, greatly complicating accurate calculation of contractile function. To address this, we increase the thickness of the base and height of the post. This is stiff enough to be essentially immovable by the contractile forces exerted by the engineered tissue and serves to reduce the effective height of the post. And by decreasing that height, we increase the stiffness of the post and solve that problem in our 96-well plate platform. So next, we cast the HTs in this 96-well plate platform and assess tissue contractile function over 28 days via microscopy. The tissue twitch force increased from 53 micronewtons on day 7 to 73 micronewtons on day 28. This shows highly functional tissue formation in the 96-well plate platform. Moving back to the actuation system now, cell-free fibrin tissues were tested for actuation limits in this 96-well magnetic plate, post format. Over here, we can see cell-free fibrin tissues were magnetically actuated, increasing magnetic fields from 1 to 10 millitesla. And in the videos on the right here, we can see the post's displacement linearly increases up to about 4 millitesla. After 4 millitesla, the deflection is again no longer within that linear region, and this deformation would complicate any downstream studies. And these results are going to be used as a baseline for tissue mechanical actuation protocols. Next, we also mentioned earlier that we were interested in transitioning our tissue biofabrication from manual to robotically controlled and automated fabrication methods. We have been able to program and use a pipetting robot for casting of tissues in a 24-well plate, and we're now optimizing casting in a 96-well plate, as you can see in this video here. So they just lifted up the cells and the extracellular matrix components. They are putting it into the wells in our 96-well plate platform. Lifts out, goes back in to mix it a little bit. And then it'll make sure it's fully clear and dispose of the tips and move on to the next set of wells. So we're also interested in enabling multimodal analysis of both contractile and electrophysiological function in our platform. To do this, we tapped on the expertise of our collaborator, Dr. Todd Herron at the University of Michigan. Todd is fully responsible for the data on the next couple of slides. Just to give a little bit of background here, he's previously developed a high-content optical mapping system capable of analyzing both calcium transients and voltage in his EHT platform, which you can see here. He's also developed a 96-well plate optical mapping system. In A, we can see a heat map for the observation of action potential duration at 80%. This system automatically identifies outlier wells and wells that fail to produce data. This allows for easy plotting of the average action potential morphology in selected wells as seen in B. And in C, we see a time-space plot generated from the horizontal line shown in A across that section over a period of 10 seconds. This system also enables high-throughput evaluation of electrophysiological drug responses. In A, human-induced pluripotent stem cell-derived cardiomyocytes are treated with isoproterenol and show increases in beat rate, both in the heat map as well as in the paired t-test. In B, we show heat maps of cells before and after isoproterenol treatment, showing inotropic effects of beta-adrenergic stimulation via both visual comparison of the heat map as well as in that paired t-test to the right. And finally, in C, we can see lusitropy, another canonical effect of beta-adrenergic stimulation confirmed with the paired t-test. This system has also been commercialized through Curie Bio. Again, shameless plug if you want it. So thanks to the groundwork and further collaboration with Todd Herron, we next wanted to identify whether we could accurately measure contractile and electrophysiological function simultaneously in EHTs. To do this, we cast some more tissues and used the Calbright 520 calcium indicator and analyzed the calcium flux and magnet position via confocal microscopy. Luckily, we found that the calcium transient precedes the contraction, which you can see in this zoomed-in plot here, sufficiently to enable these simultaneous measurements with limited motion artifacts. We also used E4031 to induce arrhythmias in these tissues, which we confirmed via both contractile and electrophysiological measurements. So here's a schematic illustration and an image of how we combined these optical mapping and mechanical sensing platforms. So basically we have our magnetic sensor circuit down here, we put our tissues on top of this, then we have LEDs at an angle to the plate used for excitation, and a camera fitted with dual emission filter placed above the plate to record that emission. This system allows for simultaneous measurement of force kinetics in all 96 wells, as well as optical mapping of calcium transients in 36 of the wells at a time. We also developed biomaterials for improved cardiac tissue function. Cardiac tissues are electroconductive, so we developed a hybrid electroconductive hydrogel that consists of decellularized extracellular matrix, or DECM, derived from porcine myocardium, and reduced graphene oxide, or RGO, a bioinert electroconductive material. Via electron microscopy, we've confirmed presence of the RGO flux in the walls of the DECM, so theoretically these should enable better electroconduction throughout the tissue. And by doing so, we were able to generate engineered heart tissues with enhanced maturity as indicated via increased twitch forces and sarcomere length over a 1-month period of time. So just to explain the data points here, we did collagen tissues, we did DECM tissues, we did our DECM RGO tissues, as well as DECM with transglutaminase. So RGO increases the stiffness of the tissues, so we use transglutaminase to cross-link it as a stiffness control for our reduced graphene oxide tissues. And we see throughout the entire period of time that we measured these, statistically higher twitch forces in the DECM RGO, as well as increased sarcomere length. We also found improved electrophysiological function and enhanced mature gene expression, which we have the biomaterials paper published last year if you'd like to look more at this work. So you like space biology. We do have some of that too. So of course these platforms and materials are developed to enable research into interesting biological questions, some of the more exciting of which are space biology works. In 2020, we successfully launched engineered heart tissues to the International Space Station to study the impacts of spaceflight on engineered heart. We can see that, again with BioServe actually, we developed spaceflight culture hardware that enabled this work. And we have astronaut Jessica Mair performing a media swap on the ISS here. And this work's been highlighted by the media previously. And in this spaceflight, we found decreased cardiac tissue function resulting from spaceflight. So twitch forces decreased significantly over time, and they did not return to baseline upon return to normal gravity here at day 39. And we found that the mean change in beat interval, a metric of arrhythmia, was increased for the majority of the time in space and returned to baseline upon return to normal gravity. Our prevailing theory was that mitochondrial dysfunction leads to this cardiac dysfunction, and we indeed identified mitochondrial dysfunction in our spaceflight samples. So first on the left here, we can see lipid accumulations next to our sarcomeres, and these are very pathological levels of lipid accumulations within our cells. In our next images, we can see rampant mitophagy and mitochondrial ultrastructural defects such as paracrystalline inclusions, concentric laminated bodies, and compartmentalization within our mitochondria. We've also used these platforms to study the effects of galactic cosmic radiation, specifically simulated galactic cosmic radiation at the NASA Space Radiation Laboratory. A lot of this data is still quite preliminary, but we're excited to share it so far. Just please don't scoop me, because then I'd have to restart my PhD, and that would make me very sad. So first we identified increased ROS presence in the tissues. So as soon as 24 hours afterwards, we have a dose-dependent increase in fluorescence intensity of our cell ROX dye, an indicator of ROS. This trend is maintained, though without statistical significance over time, up to day 30. We also found increased arrhythmias in our cardiac tissues, again using the interval irregularity as a metric for arrhythmia. As soon as 24 hours following exposure to simulated galactic cosmic radiation at 25 centigrade dose, we have increased interval irregularity, and this is further exacerbated with time, as you can see from that 7-day time point. We also found pathological mitochondrial fragmentation 1 week following exposure with 25 centigrade GCR-SIM. This correlates with previous work that's identified oxidative stress and mitochondrial dysfunction as a central hub for spaceflight-induced tissue damage. We've also started to study the effects of simulated microgravity on skeletal muscle tissue myogenesis. So C2, C12 skeletal muscle cells were cast into tissues and then placed on the random positioning machine, which you can see here. So a random positioning machine attempts to simulate microgravity by randomly moving the sample in 3 dimensions, as the name random positioning machine implies, such that the average gravitational force vector sums to 0. Interestingly, the average myotube size is decreased in both diameter and length when formed on the random positioning machine. And as you can see, if I can get these videos to play, which would be quite nice. I think okay, it seems quite laggy, but it appears to be working reasonably well. The twitch forces under pacing are also decreased in case that video is actually malfunctioning. We did quantify this, and we found decreased twitch forces and tetanic forces in our skeletal muscle tissues when cultured on the random positioning machine. And finally, this may be explained by our Western plot data that shows decreased expression of both slow and fast myosin heavy chain isoforms. So finally, I'd like to thank everybody in the lab, especially Dako Kim, who wasn't able to make it here today, as well as a lot of the people that did the work responsible here, especially Guangjun. He did all of the mechanical actuation work and is a fantastic postdoc in our lab, as well as our collaborators, funding sources, et cetera. We're also excited to promote the upcoming International Conference on Biofabrication in PISA this fall, and abstracts can be submitted online. And then finally, we have postdoctoral and graduate student positions available, so please reach out to .co at this email here if you're interested in joining some of our work. With that, I was only tangentially involved with some of the work I presented today, so I'd be glad to try to answer any of the questions you may have, or at least direct you to the correct resources for getting your questions answered. Thank you.